Hi, my name is Tatyana Zamkovaya from Anna Knesset's lab at the University of Florida, and today I'll be talking to you about a computationally intensive approach to discover novel adaptation genes in extreme environments. So the reason why this is important to study is because in order to better understand how life evolves and adapts to extreme conditions within and beyond Earth, we really need to better understand unknown taxa, also called micro-dark matter, that dominate even the most extreme of environments, but whose ecological and functional relevance remains unknown. In our previous work, we conducted a network-based analysis to uncover the ecological relevance of unknowns. We collected over 250 samples from all over the globe from publicly available data for 4 different extreme environments: hot springs in red, hypersaline in green, deep sea in turquoise, and polar, Arctic, and Antarctic in blue. We then constructed networks for each of these individual environments, and we used the network measure hub score to identify the most important taxa. In our case, taxa with the highest hub score would be the most prevalent and most connected or abundant taxa within each environment, and would also be the taxa most essential for maintaining network structure, shape, and integrity, as when these taxa are removed, many connections are lost. So what we see here is bacterial networks at the genus level, where all nodes are colored by genus, with unknowns being represented in gray. And all nodes are sized by hub score, with a larger node size indicating a larger hub score. What we can see is that unknowns predominantly act as top hubs within these extreme aquatic environments. We hypothesized that due to their integral network position, these extreme environmental hubs may therefore possess optimized adaptation strategies, and therefore are ideal candidates to search for novel biological pathways of survival. However, because we were working with amplicon data before, we couldn't really investigate their functional role unless we did a novel computational approach. In fact, we decided to use 16S hubs as probes to find novel genes within functionally annotated metagenome scaffolds, thereby bypassing existing challenges like the lack of well-annotated reference genomes for unknown taxa. In fact, our Hublas pipeline begins with the 16S RNA FASTA sequence of each hub that we blasted against a large curated metagenome database that we collected and created by collecting all the publicly available metagenomes found on the ISMB web server to make a blast database that amounted to 13 terabytes in size. When we blasted against our metagenome database, we wanted to find scaffolds that not only had a high percent identity match to the 16S query sequence with at least 95% identity or above, but that also had a high number of genes with at least 30 genes to allow us to identify as many novel and interesting genes as possible. In order to figure out what kind of genes were present in each scaffold, we have 2 main resources: annotation databases like KEGG and PFAM, and a list of known adaptation genes that we created by text mining for adaptation terms within literature, finding genes with functions related to stress, survival, and adaptation to extremophilic conditions. We then looked for putative adaptation operons, in which at least 3 or more genes with functions related to stress or survival, in this case to heat shock in red, are located alongside a novel gene with no known functions in black to help us figure out the function of these novel genes. We can also use other tools such as a sequence alignment or protein interaction network to further characterize these unknown genes. In fact, we came up with 3 main strategies. The first being to perform function prediction of hypothetical genes within a selected operon. The second, to do a similarity analysis to find highly conserved hypothetical genes. And the third, to do a comparative phylogenetics and function prediction analysis by blasting all hypothetical genes against our metagenome database to find not only highly conserved hypothetical genes, but also highly conserved operons, regardless of the environment they first came from. When we look at our results, we see that our approach was successful. It helped us find high-confidence gene-rich scaffolds for both unknown and known hubs belonging to different environments. When we initially set out to do our analysis, we wanted to find novel genes related to adaptation from unknown hubs, but we also wanted to use known hubs as a control. So we blasted at least 20 hub sequences for known and unknown gene genera for each environment. And what we found is that when we look at the average percent of hypothetical genes, adaptation genes, and mean operon counts, we see a similar and high number, regardless of hub or environment type. We then performed function prediction using our different strategies. We found that function prediction was possible and successful when examining select hypothetical genes in scaffolds. And here we show one example where we looked at hypothetical genes in yellow from a hot spring scaffold, and we found that these hypothetical genes most likely have oxidative stress-related roles based on their proximity to genes like buprenorphine and iron sulfur oxidases. We also found that function prediction was possible and successful in a large-scale manner when we blasted 2,600 deep sea and hot springs hypothetical genes using a sequence-sum learning network of an all versus all blast. We found that hypothetical genes, regardless of which environment they originally came from, clustered well with another based on their superfamily origins. We also performed a sequence-sum learning network of an all versus all blast of hypothetical and uniprot DEF3035 genes, where our hypothetical DEF3035 genes from our analysis are in pink, and all the uniprot genes are in purple. And when we look at the network, we see that these sequences clustered well with another, indicating high similarity in sequence. And when we performed a phylogenetic analysis, we saw high phylogeny as well, where our hypothetical DEF3035 genes are phylogenetically similar to other uniprot DEF3035 genes, specifically from Brevindomonas. So in conclusion, our approach enables identification and prediction of novel genes, and it is also highly accommodating for the computational power and memory required of large-scale datasets. We found that function prediction was possible and successful for select operons and in a large-scale manner, allowing us to identify conserved and highly similar hypothetical genes. In future, we hope to identify more survival stress genes. And we'd like to thank all of you for listening and our grant for funding this project. Thank you.